Well, you're on. Stress highs? That is not a good thing to have. It's better than non-stress highs, just natural highs. Oh yeah, you know, well, is she actually have anything coming up after this stuff? I love when professors are normal, living human beings that are just like, I don't want to drive through this, I'm not going to make you drive through this. And then there's the others that are like, yes, you must trudge through, like, the cold, bitter winter. Wind chill is negative 27 degrees, yes, please. The power is out. So that was last year. Yeah, that's fine. Oh, okay. I'm like, so what? You blended with the snow in your car?
Any here yet? Rosa? Emily? Jesse?
Then how are you not going? You would have said it, but I would not have if she would have tried. Hey, I have perfect It wasn't terrible though. It wasn't terrible.
When we had to draw the starting material, I was like, oh damn, here we go. When I saw that there was 4 of them, I was like, I did.
Difference between these two, this one, was this kind of go?
Went alright. I finished in like 30 seconds.
Changed something.
Now
it looks like this.
I mean, it was okay. It wasn't great. I thought the multi was really good. It required a lot of thinking, but like it wasn't like it.
It's just changing like some.
But this was her accent.
I went to Vietnamese and
That was radical. I put it as radical initiator. That seems like a trick question.
I did on my phone too. This is kind of cool.
I never knew it was called a poison. Did I have poison or something? Well, there's
Actually, so this,
Yeah. Yeah.
Candace got a brain injury.
I did, when I clenched my jaw so hard that it hurt. I was hoping that I do fall and get double summer.
Oh, yeah, reading those.
It's like one of my favorite songs to listen to when I was. I used to like read them. Why?
Yeah. Really? Yes. Right. I'm just still not understanding why we're
Oh yeah, she sent me a voice memo and I go, Debbie, I think you're talking about something else, and she goes, oh no. And I was like, I'm still don't understand what the difference is. And she's like, Rose, I'm just confusing myself by talking with you. We're just gonna take the L. I have class E3, work at 4, e-board meeting at 9. I can't go home yet. It's because so I'm wondering we already did with these coins. He's going. Don't worry, we already.
What? It doesn't hurt, and then it's still.
Just can't imagine having to be in a group for that long. It's just rough.
Last year I was in it for 6 months, and I broke my metatarsals.
Oh my god. You just live in a booth?
Yeah.
It's like you and Barbara and the caretaker. Barbie would.
Wait. I didn't realize you personally 500. 500. Yes, 500. How much did you sell? 500. And they were like, well, it'll be okay, we'll take care of it. No, I'm just gonna have to pay out of pocket. But everything. I'm gonna try to go in there. Yeah.
Does it like, do you like file things?
Maybe like, where do I want to save it?
Yeah, I tried that.
I think it'll be okay because I'll have
I got nothing after this week or at the board.
Hey, Henry. All right, guys, good morning. Take quick attendance. Riley,
Here, I'm getting my list for you.
Brianna, Drew, Bria, Henry, Anna, Nick, Abby, Hannah, Rosa, I have your exam to pick up. Matt, Jillian, oh. Mia, Annie, Brianna, sorry. Brandon, Sadie, Jessica, Jesse, William, Marissa, Anna, Sophia, Aubrey, oh, sorry, couldn't see you. Matthias, Colin,
Olivia? Braden?
Here.
Braden and Brandon, you two are the ones that I keep getting confused because your names are so similar and you sit next to each other. You're Braden, right? You're Brandon. Got it. Amber. Here. Katie. Ben. Kennedy.
And then she said she just.
Ava. Kendall.
Here.
Emily. Here. Lauren.
Here.
Jennifer.
Here.
And Isabel. And Corwin. All right. Okay, guys. So worksheet 3 is going to cover chapter 6 and chapter 8. I already have posted it, but you're not going to be able to answer all of the questions on it because the last chunk is on the next part that we're going to talk about on Friday, but you're welcome to get started on it. It's a longer worksheet, really in-depth, helps really cover everything that we've learned so far. So you're welcome to get it started, but it won't be due until Monday. So you guys we'll go through the rest of chapter 8 on Friday. Okay? So today we're going to continue with this part of the lecture on transcription and translation. Any questions on any of the steps of transcription? We talked about initiation, elongation, and then termination, and the 2 types of termination. Any questions on any of that? Okay, well, just to remind you, for termination of transcription, there are 2 processes that can occur. One is dependent on the row protein, and one is not. This one is dependent on when the hairpin gets formed and then the RNA polymerase contacts it, and essentially there's a knot in the template, and so you're unable to continue on with the transcription. And so up until now, we've talked about these mRNA transcripts, which are essentially the messengers that will make proteins by the use of ribosomes. But what I'm showing in this chart is that there are actually several other classes of RNA, and each is designed for a different purpose in the cell. So our RNA forms the majority of the ribosome, so it's all RNA. So ribosomal RNA, that's what rRNA is. It's part of the ribosome, majority of it is the ribosome, and it also includes the ribosome's catalytic center, which we'll talk about. It's called a ribozyme. So anything that has catalytic activity is an enzyme, so a ribosome that's an enzyme is termed a ribozyme. tRNA or transfer RNA is one we're going to talk about. So it basically shuttles the amino acids to the ribosome to then synthesize the protein. There's small RNAs or sRNAs or regulatory RNAs. These don't encode for any protein, but they can directly help regulate transcription and translation. We're not going to get into too much detail with those. tmRNAs are properties of transfer and messenger RNA, also pretty unique, but all you have to know is that essentially, they're a type of RNA with a certain purpose. You don't have to get too much in depth on this, but they do help ribosomes that get stuck on damaged RNA. And then, like I said, there's the catalytic RNA, which can carry out enzymatic reactions. So if you see a specific type of a ribozyme called RNase, remember anything that ends with SE is an enzyme. So that's an RNA, that's also an enzyme. That's a specific type of ribozyme. Does that make sense? Now, notice here the dramatic differences between these half-lives. So this is essentially how long it takes for that molecule to be degraded in half. So the average half-life for mRNA is about 3 to 5 minutes. Some can be 15 seconds, others can be 30 minutes. But this really is telling us about the stability of these different kinds of RNAs. So if you look at the tRNA and the rRNA, tRNAs take hours, RNA also takes hours, so they're a lot more stable. And the reason for that, which you guys should know, is that these tRNAs and RNAs, if they have a higher half-life, they're more stable, and this is because they have certain modified bases that are less susceptible to RNase digestion, or digestion of the RNA enzymes. So they're less likely to be degraded. And there's a reason for these having a longer half-life versus the mRNA. And it'll start making sense as we go on. Any questions on this? Okay. Now, concept check. See if you guys are paying attention. The termination of transcription can be caused by which of the following? Yes.
Is the stem loop the same as the hairpin loop?
Yes.
Then
Sorry?
D.
D, yes, that's correct. A and B are correct. So the stem loop structure is essentially, you're right, the hair loop structure, and then the row factor. So it can be row dependent or row independent. Does that make sense to everyone? All right, cool. So quickly, we'll go through some general biology here. Once a gene has been copied into mRNA, so that's transcription, the next stage is translation, which is the decoding of the mRNA to synthesize the protein. An mRNA molecule can be thought of as a sentence in which there are triplicates of nucleotides that are called codons, and those codons represent individual words, which are essentially the amino acids. So we put together codons that code for an amino acid, and all of those put together makes a sentence, which is the whole mRNA molecule. That final sentence that you make is the polypeptide sequence. So ribosomes essentially are machines that can read that mRNA language and translate it into protein. And they do that by the genetic code. So across all three domains of life, we use the same genetic code. All of life, literally with very, very, very few exceptions, uses that same genetic code, whether you're a human or you're a phage. So the genetic code consists of these nucleotide triplets called codons. So if you remember, codons are these A's, C's, U's, and G's, because right now we're talking at the mRNA level, so we have uracil instead of thymine. There are 64 possible codons that you saw on that chart. 61 of them specify amino acids. That will also include the start codon. And then there are 3 stop codons. So in this question over here, C is a stop codon. Could have been a trick to for you guys, but remember now we're talking about translation, and translation is the one that has stop codons, not transcription. So try to keep these things straight in your head. There are 3 different stop codons, but the genetic code is degenerate or redundant, which means that you can have more than one codon encoding the same amino acid. So what that means is you can see that the amino acid leucine actually has 6 different codons that can specify for the amino acid. So just to remind you guys, each one of these is a nucleotide, but when you have 3 together and those 3 can specify an amino acid, then it's called a codon. So these are all the different amino acids, all the codons. AUG, or methionine, does anyone recognize this one? It's the start codon. Yep, good job. So the 3 stop codons are over here, UAA, UAG, UGA. This is a start codon, and everything else codes for amino acids. So does AUG. It's the amino acid methionine, but that's the start codon. Okay? So you need to know, in terms of memorization, just the start codon. You don't need to memorize all of the amino acids. That's something you do in biochemistry. Questions on this? So now at this step, we have these codons, and we're at the mRNA level. We have our mRNA sequence. So now we have the transfer RNA, which are these molecules that decode and convert the language of the RNA into proteins. Essentially, they're like an adapter molecule that can convert that codon alphabet into an amino acid alphabet. So these tRNAs essentially are just floating around the cell, and they have to get charged with the right amino acid, and then they can return to the ribosome and drop off that amino acid cargo. So they're essentially transferring the proper amino acid to the ribosome. They have to get charged in some way by it, and then they take it to the ribosome, and the ribosome puts it together in a chain of amino acids to make the proteins. Because remember, amino acids make up proteins. So you don't just convert the language of mRNA into protein, there's steps in between, and this is where the tRNA comes in. So there are 3 functional regions of the tRNA. The first is the anticodon, which is right here, and essentially this hydrogen bonds with the mRNA codon. So this is the mRNA, AUG, the start codon, and then you have UAC, which is essentially the complementary, that's the word I'm looking for, the complementary basis for the mRNA. There's also the 3' acceptor end, which is up here. And so this is the part that binds the amino acid and brings it to the ribosome. And then there are these side loops. And so these are essentially recognized, this is the part that is charged with the right amino acid that is then matched to that particular anticodon. So this is essentially, that's the 3' acceptor end. This is the part that's charged with the amino acid. But the side loops are just modified bases that help protect this from degradation. So tRNAs, they have a lot of pretty unusual modified bases that have, like these loops that just help them from being degraded, and they're kind of just floating around in the cell waiting for, the right amino acid to charge them so that they can bring that amino acid to the ribosome. Does that make sense? Any questions on that so far? Okay. So once we have these charged tRNAs that are floating in the cytoplasm, they bring amino acids to the ribosome. And so the ribosome can then translate that mRNA into these amino acid sequences of proteins that have some function in the cell. So ribosomes are composed of two subunits, each of which contains rRNA and proteins. Remember I said that RNA is ribosomal RNA. It makes up the majority of the ribosome. The other component is just more proteins. So in prokaryotes, there is two subunits. There is a small subunit and a large subunit. The small subunit is called the 30S subunit, large is called the 50S. Together, they make the 70S ribosome. So the full holo ribosome enzyme is the 70S ribosome. This might confuse people because 30 plus 50 is 80, but it has nothing to do with adding the two. It has stuff to do with other things. So we don't need to get into the details of that. It's a different unit in like Spedbergs or something. So it's not additive. But they exist separately and then come together on an mRNA transcript to form the full translating ribosome 70S. So the tRNA brings these amino acids to the ribosome. The ribosome has to stitch them together to make that polypeptide sequence. So the ribosome makes those peptide bonds that stitch those amino acids together, and the enzyme that it has is called peptidyltransferase. So the ribosome is a ribozyme, and that peptidyltransferase is the enzyme that makes it a ribozyme. It's essentially an RNA molecule that carries out catalytic activity or enzymatic activity. This ribozyme is part of the large 50S subunit. So while this ribosome is highly conserved across different species, there are certain differences in our RNA sequences that obviously increase in relation as species are more similar to each other. But in blue, we have the rRNA, the catalytic center. And then in yellow, you have these other proteins. So the protein actually largely plays more of a structural role, not an enzymatic role, because that catalytic center is what's really made of those ribosomal RNAs. Given the importance of RNA in translation in bacteria, as you might imagine, the sequence of this rRNA, the ribosomal RNA, is pretty similar across different species and different microbes, just largely because of how important it is to translate protein properly. But there are very minor differences. And so as a result, it serves as this molecular clock for evolutionary biologists because it's a way to measure the time in which two species have diverged from one another. So you can look at this part of the RNA, and so often when we want to identify an organism and whether it relates to another bacteria, we usually go for the 16S rRNA. You guys have probably heard this. It's a sequence that helps you determine what it might be, because all bacteria are going to want to translate mRNA into protein, so we go for something that they'll all have, this rRNA, and there's only those small differences between different bacterial species. to help us identify it. So in eukaryotes, it's an 18S rRNA. I think first lecture when I was comparing prokaryotes to eukaryotes, I mentioned this, but this is why. Any questions on this? The 70S ribosome, so the whole thing together, has 3 binding sites for tRNA. So remember, tRNA is the one that transfers the amino acids to the ribosome. The tRNA has to bind to the ribosome. There are 3 sites for that to happen. An acceptor site, it binds that incoming amino acyl tRNA. There's a peptidyl tRNA, or a P site, that harbors the TNA with the growing polypeptide chain, and then there's the exit site that then binds a tRNA that is recently stripped of the polypeptide because now the ribosome has taken it and converted it to an amino acid. So during translation, every tRNA molecule has to move through these 3 sites in the ribosome, sort of in this assembly line fashion. It enters through the A site, the acceptor site, the charged tRNA. When it's charged and it binds, we call that the amino acyl tRNA because it's been charged essentially. And then it moves through the P site where the chain starts to grow. And then the amino acid is added to the polypeptide chain. You're growing that polypeptide chain. And then once it's uncharged and it's been stripped of its amino acids, then the tRNA can kind of float off and exit. So this is an image of a ribosome. The mRNA is in light blue. The red is the 50S subunit, the pink is the 30S subunit. Together they make the 70S whole ribosome. In the center, you have in blue, which is the acceptor site. In green is the P site, and the exit site is in yellow. So they're in order, going from right to left. So the acceptor site, basically the mRNA is right here. And so you have the A site in blue. This is where the tRNA comes in. It gets charged, and then it's able to pull the mRNA through and start adding these polypeptide sequences in yellow. And so as it does that, through the green site, the P site, it eventually will run out of those amino acids that it's carrying for the ribosome. And then when it's stripped of that polypeptide, it becomes uncharged, and then it gets out through the exit site. Does that make sense to folks? Okay. So we've gone through transcription, translation, but now let's talk a little bit about where they happen in the cell, and why that matters for the cell. So can anybody remember one of the key things that we talked about in prokaryotes that has to do with transcription and translation? You guys were tested on this? Yeah. Yes, they are coupled. They occur simultaneously. So now you have to kind of zoom out and think about it in the context of the actual bacterial cell, right? There's DNA there. There's in the nucleoid region. And then, this is all the cytoplasm. There's ribosomes that are floating around in the cytoplasm. So you can be making essentially mRNA and then have a transfer RNA come and grab those ones that it's making and take it to the ribosome. The ribosome starts to make protein while at the same time there's more mRNA being made. So that's why bacteria can really grow and double really quickly as well. So before the RNA polymerase can even finish making that entire mRNA molecule, ribosomes can just start binding and to the end of that mRNA and start translating protein. So this is called coupled transcription and translation. Since there's no nucleus, that allows the ribosomes to immediately bind because the DNA essentially isn't enclosed in anything. So the ribosomes can bind and start making, proteins before that mRNA is even done. So it's simultaneous. Eukaryotes, they have a membrane enclosed DNA, it's called the nucleus. So they have to use separate compartments to carry out most of their processes. So transcription happens in the nucleus, because that's where the DNA is. If you want to convert it to mRNA, you got to get into the nucleus. Translation occurs in the cytoplasm, that's where the ribosomes are. Does that make sense? Okay. Now, polysomes essentially are a multi-ribosome structure. So once a ribosome begins translating mRNA, another ribosome can then immediately jump onto that ribosome binding site, and the mRNA can actually have more than one ribosome moving along its length at the same time. So when that's happening, so here we have DNA being converted into mRNA here in the blue, and so you have one ribosome making this protein sequence, another ribosome is also just right behind making more protein. So this, ribosomes that are in a polysome essentially are usually closely packed along the mRNA. And another positive reason for this evolutionary, mechanism is that they protect the mRNA from RNases, that things that could come in and try to degrade it, it's not going to be able to do that because there's ribosomes doing work in that factory, essentially. So they also speed the production of protein from a single transcript. So what's useful about this is that they can rapidly translate proteins under that particular condition, and having multiple ribosomes on that transcript actually helps its stability and helps it from being degraded. So once again, just another way to say this, but remember, we're talking about stability again. And in the very beginning, I talked about, the stability being related to half-life. And so this is kind of where you can connect the two. When you are dealing with the ribosomal RNA and the transfer RNA, those are essentially going to be more stable and have higher half-lives because of that catalytic activity and that potential for more than one to be working on an mRNA transcript at the same time. So that helps protect them from degradation, which helps with their half-life. Does that make sense? Okay. I'll leave you here. You get to leave a little bit early, but I'm really doing this because this is a lot to digest. It might make sense now when I'm saying it, but if I were to quiz you on it, it's going to be difficult to just recall from memory. So please take the extra time to study this because we're going to just keep moving on Friday. Okay. If you haven't picked up your exam, I have it here. I have Rosa and Annie. What? Is